Thank you. My name is Kazuto Kimura. I'm with Osaka Metropolitan University. Today I'm going to talk about red blood cell differentiation using canine iPS cells. This is the outline. I'll focus on the red blood cell differentiation protocol using canine iPSCs and the differences in differentiation efficiency among cIPSC lines. Let me start by describing the background. In veterinary medicine, no blood bank systems have been developed. So we have no choice but to rely on blood donor dogs for transfusions. But their availability is severely limited. For this reason, blood shortage is a serious problem in veterinary medicine. In contrast, canine iPSCs have great potential to provide a continuous supply of large numbers of red blood cells for transfusions because they have self-renewal and pluripotency. We have reported an efficient reprogramming method for our blood cells into canine iPSCs. By applying this method, we were also able to generate canine iPSCs from other cell types, including skin dermal fibroblasts and erythroid precursor cells. Furthermore, we developed an efficient canine iPS cells culture method. So currently, we have developed canine iPSC generation and culture methods. However, we have no information on canine iPSC RBC differentiation methods. In addition, to apply canine iPSC RBCs to blood transfusions, a large number of RBCs must be produced. So we investigated the efficient cIPSC RBC differentiation method. In human or mouse iPSC research, some studies have suggested that blood cell-derived iPSCs could have better differentiation potential into hematopoietic lineage than dermal fibroblast-derived iPSCs. In other words, donor cell type can influence the differentiation potential of iPSCs. So we hypothesized that erythroid precursor cells would be a superior cell source for iPSC RBC compared to PBMCs or dermal fibroblasts. So one of the main goals of this study was to develop a canine iPSC RBC differentiation protocol. Additionally, to test the hypothesis, we compared the cIPSC RBC differentiation efficiencies of canine iPSCs derived from multiple cell types. Now I'm going to go on to the materials and methods. Dissociated canine iPSCs were seeded in 96 well-U plate to form spheroids. The following day, the spheroids were transferred to cell culture plates and cultured in hematopoietic differentiation medium for 24 days. Here is the protocols. The culture conditions, including the media and cytokines, were changed as you can see. The cells obtained during the differentiation process were characterized for morphology, marker expression, and hemoglobin expression. Let's look at these results. On day 6, you can confirm a few spherical cells. The number of cells increased from day 6 to day 18. This slide shows the morphology of the Gimza-stained cells. You can see many polychromatic erythroblasts on day 12 and orthochromatic erythroblasts on day 24. These red arrows show a few enucleated RBCs. Next, I want to look at the pluripotency markers expression. The expression of these 3 key pluripotency markers decreased with differentiation. This means canine iPSCs can be differentiated using our protocol. This is the differentiation markers expression data. From days 0 to 6, mesodermal and early hematopoietic marker expression increased, and by day 9, the expression almost completely decreased. At the same time, we observed hematopoietic and erythroid marker expression. All markers expressed during differentiation process. So these data tell us the protocol can differentiate canine iPSCs into erythroid lineages. Next, I'd like to look at the hemoglobin expression data. These are pictures of differentiated cell pellets on days 9, 12, 18, and 24. You can see that the cells look red, and the red color is getting dark with differentiation. This slide shows the hemoglobin gene expression. As shown in the white box, cultured erythroid precursor cells and bone marrow mononuclear cells mainly expressed adult type hemoglobin. In contrast to these adult cells, the yellow box shows the band of differentiated cells from canine iPSCs on each differentiation day. You can see the differentiated cells expressed all types of hemoglobin genes, including the embryonic, fetal, and adult types. Here is a brief summary in section 1. We were able to develop canine iPSC erythroblast and RBC differentiation protocols. So in the next section, I'd like to talk about the differences in differentiation efficiencies among canine iPSC lines. This slide shows the materials and methods. We used 4 canine iPSC lines derived from erythroid precursor cells, PBMCs, and dermal fibroblasts. Each canine iPSC line was differentiated into RBC using a previously described protocol, and their differentiation efficiencies were compared. The efficiencies were characterized by morphology, cell number, pluripotency, and differentiation marker expression. First, here is the result of morphology. Erythroid precursor cells and PBMC-derived canine iPSCs were able to produce many spherical cells, but a few spherical cells were differentiated from dermal fibroblast canine iPSCs. On day 24, erythroid precursor cells and PBMC canine iPSC-derived cells looked dark red, whereas dermal fibroblast canine iPSC-derived cells looked light red. This slide shows the cell number. The number of differentiated cells derived from PBMC canine iPSCs was the highest, followed by erythroid precursor cells and dermal fibroblasts. Next, I look at pluripotency marker, SSEA1 expression. These 4 different colored graphs represent each canine iPSC line. The expression rates declined with differentiation in all canine iPSC lines. This figure shows the percentage of mesodermal marker KDR positive cells. On day 3, PBMC canine iPSC1 line had the highest rate of KDR positive cells compared to all other canine iPSC lines. This is the CD34, which is hematopoietic marker expression data, as well as the result of KDR expression. PBMC canine iPSC line 1 had the highest percentage of CD34 positive cells than all other canine iPSC lines. Here, I would like to summarize the main results in this section. Contrary to the initial hypothesis, the PBMC canine iPSC line, especially line 1, but not the erythroid precursor canine iPSC line, differentiated efficiently into canine iPSC RBCs. This probably means that the selection of canine iPSC lines, which have high RBC differentiation efficiencies, can enable to produce large number of canine iPSC RBCs. I'd like to discuss remaining issues in this research. In section 1, I talked about the canine iPSC RBC differentiation protocol, but there are challenges to overcome for its application to transfusions. One is the low inclusion rate. This can put us at risk for tumor genesis and immune rejection after transplantation. Another is that the hemoglobin types of canine iPSC RBCs include embryonic or fetal types, which may differ from adult RBCs in their oxygen binding capacity. In section 2, we have found the differences in RBC differentiation efficiencies among canine iPSC lines, but we don't know whether the donor cell types of canine iPSC line truly cause differences in differentiation potential among the canine iPSC lines because we only compared a small number of cell lines. So to clarify this question, more experiments using many cell lines are needed. So in conclusion, we have developed a canine iPSC RBC differentiation protocol. However, issues remain regarding the low enucleation rates and embryonic and fetal hemoglobin expression. We also found that RBC differentiation efficiencies differed among the canine iPSC lines. This probably means that the selection of great canine iPSC lines can improve RBC differentiation efficiency. Finally, I want to thank the people involved in this work. Thank you for your attention.
Thank you very much. Any questions? Any questions? Yes.
Thank you, Kazuto. Excellent presentation. Do you have any thoughts how to potentially, it looks like your cells, just looking at them, they really do they look like erythroid precursors. Do you have any thoughts? How can you promote, the final maturation of those cells?
Oh, it's a difficult point for me, but I think the culture conditions are very important for enucleation because the enucleations is suggested to be caused by the blood flow in vivo. So I think the 3D culture and the bioreactor or something like that can improve enucleation rate. It's one thing.
Is there any need in erythropoietin supplementation? EPO, the enzyme that is necessary for hematogenesis elsewhere. Are you providing it in your culture?
Sorry, one more.
Are you providing erythropoietin in the culture?
We added erythropoietin. Yeah, thank you.
All right, last question.
Well, regarding the efficiency for the differentiation, what made you to terminate the differentiation at 24 days?
I referred the human iPSC RBC differentiation protocol, and the protocol is 24 days protocol. So I just tried the same conditions. Yeah, more culture can improve some maturation rates or something, I think.
Thank you very much.